interesting question. I have spoken about this in the past, but I have about 5 minutes to wait for an incubation, so I thought that I would answer your question anyway. So, first thing that we need to consider is that my lab coat isn't going on. Okay, here we go. So the first thing that we need to consider is that there are different ways that brains are processed. So there's 3 different feels I'm going to describe in this video. So firstly, a fresh brain coming straight out of a human's head is very, very soft. Closest thing I can think to describe it as would be ripe avocado. So you give it a prod, really, really soft. And it's important to note as well, your brain inside your head is supported by cerebrospinal fluid. It's basically floating in a bath. And this supports the structure, this keeps its structure as it is. But if you take a brain out of a head and just pop it on a table, it will just sink down. It can't support its own weight. So it's very, very soft. That's probably the craziest feeling. And I think your question is probably alluding to a specific case, which I will get onto later. But in terms of what different types of brain feel like, I thought I would answer those first. So when a brain bank receives a brain, usually 2 different things are done. So the brain will be chopped sagittally, so this way, down the middle, and separated out. So this side, the right-hand side, is usually frozen. It gets chopped up into slices and then frozen at minus 80 degrees. This is really useful for preserving the DNA, all of the genetic material. What it's bad for, though, is it doesn't really look like a brain under a microscope anymore. It doesn't preserve the morphology very well. So to sort of counteract this, the other half, the left hemisphere of the brain, is fixed. It's preserved for about 2 weeks in a chemical called formalin, and this really hardens it up and it preserves the protein in the brain. So then you can chop it up and you can look at it under a microscope, and it preserves the cellular architecture. This is really useful for diagnosing and seeing pathologies in the brain. So when we have the frozen brain, it feels much like you would imagine anything at minus 80 degrees feels, very, very cold and rock hard. But if it starts to thaw a bit, it's a bit slimy. The other hemisphere, the formalin fixed hemisphere, it sort of firms it up a bit when you fix a brain, and then it can sit on a table and hold its own weight. And at this point, it feels more like tofu, I think. I don't really eat tofu very often, but that's probably the closest thing I can think to describe it of it. If you give it a prod, it'll sort of bounce back into shape. There is a video pinned on my page of how we actually chop up the brains using a microtome to see them under a microscope, so feel free to go and watch that if you haven't already. So that's what brains feel like in their various different states. In terms of I don't want to say craziest feeling, because I don't think that's very sensitive to a real person's brain that I've felt. I've never felt an abnormal brain. Obviously, with the different diseases that I'm looking at, for example, Alzheimer's disease or frontotemporal dementia, these brains do look different to each other. The gyri, the wrinkles in the brain, are much smaller in disease cases versus healthy controls because the brain cells have died. So there is that sort of visible difference, but in terms of how they actually feel, they don't feel very different. There was, however, one case of someone who had Parkinson's disease, and they had DBS, which is deep brain stimulation, and the way that this works is that an electrode is... Sorry, someone just interrupted. An electrode is put into the brain, and essentially it stimulates the understimulated parts of the brain in Parkinson's disease to counteract some of the simple symptoms that people have. And in this specific scenario, when we received the brain, I wasn't aware that the probe was still inside the brain, so I picked up this brain and there was a bit of metal sticking out of it, which was a bit of a shock. The rest of the brain didn't feel any different, but there was this element of surprise. So I hope that answers your question. But yes, no, in general, I would never say that anyone's brain feels crazy or weird or anything like that. Brains as a whole are kind of weird things. And the textures, they're quite texturally challenging, I would say, to work with. But certainly, there's no disrespect to anyone's brains out there. That's all. Bye-bye.